Hi, welcome back. In the last module, we tried to give a picture of the problem of intractable heart failure and why it is still an unsolved problem in cardiology. In this module, we will investigate whether the heart can regenerate and if so, how. We will also look into some of the mechanisms that different animals use to regenerate the heart. Finally, we will also try to investigate whether the human heart regenerates as well and what we can do to help this process along. Before we start, let us get to know some facts about cardiac regeneration that you might find useful. Let us have an overview of the problems that face a cardiac regeneration. The heart is one of the least regenerative organs of the body. It has 2 to 4 billion cardiomyocytes, and a heart attack can wipe out 25% of these in just a few minutes or hours. Other chronic problems like hypertension can kill cardiomyocytes slowly over the years. About 20 million cardiomyocytes are lost every year because of aging, even in the absence of heart disease. The goal of cardiac regeneration is to regenerate the myocardium that has been damaged and help the heart regain functions that it has lost over years of disease. We will look into the exciting research that has been done to see whether we can pick up some of the tricks which will help us to do just that. This is the zebrafish. Originally, it evolved in the Gangetic Valley of India and the Himalayan region of the Indian subcontinent, but today it is found everywhere where there are fish lovers. It is a great favorite of aquarium keepers because the fish is cheap, breathes easily, is playful, and all in all is a lovely fish to have in your home aquarium. But it is also a great favorite with scientists. Biologists love it. It can be observed fairly conveniently and has some unique abilities, one of which we shall see in a moment, and its genome has been fully sequenced. It has been used by neurobiologists, developmental biologists, geneticists, and many others. It interests us because the zebrafish has a unique ability to regenerate its heart after it has been damaged. Coming closer home, let us look at a mammalian species like the mouse. 1-day-old neonatal mice have a similar response as do zebrafish, but this ability is lost even in 7-day-old mice. The mode of action is the same as in the zebrafish model. However, mammalian hearts do have a low level of cardiomyocyte cell cycle activity, which can replace damaged cells, and this has been known since the 1960s. This, however, occurs at subtherapeutic levels. That is to say, enough regeneration does not occur that will lead to repair of the heart and improvement of its function. But what about the human heart? Does it also regenerate? Conventionally, we would answer no. This was the stated position of all researchers until just a few years ago. This seemed obvious. Cardiologists do not see any repair process in the heart which involves new heart muscle formation. All repair appears to be by fibrosis, which leaves a scar which is often the cause of heart failure. But there are certain pointers which suggest that the heart muscle cells, the cardiomyocytes, may have some regenerative activity after all. The first suggestion that this might be true was when a group of researchers way back in 1971 did some complex mathematical modeling, the nature of which need not detain us here. Their calculations suggested that if we postulate that the number of cardiomyocytes remain the same or only decrease over time, it is not possible to account for the increase in heart size that occurs as an individual grows. They showed that the actual increase is more than would be expected by merely the increase of the size of the heart cells. Thus, they postulated there must be an increase in the number of cardiomyocytes as well. In other words, cardiomyocytes, heart cells that is, must be produced in adult life. However, there was a lot of debate about this hypothesis because the original paper made some assumptions about the heart muscle size and the DNA content which were not universally accepted. Another group took a different tack. Bergman and his co-workers took advantage of the limited nuclear test ban treaty which was signed in 1963 They utilize the fact that carbon-14 concentrations in the atmosphere sharply increased in 1955 after the above ground nuclear bomb tests and dropped exponentially after 1963 when the treaty came into effect. Humans take up carbon-14 with their food and incorporate it in their DNA. Thus, a person born before 1955 should have cardiomyocytes with low carbon-14 content if it is true that cardiomyocytes stop to proliferate after birth. Instead, it has been found that cardiomyocytes had much higher carbon-14 content and thus must have synthesized DNA in the years before or even after the increase of atmospheric carbon-14 content. They concluded that there is a very slow but steady turnover of cardiomyocytes during adult life, around 1% a year, which demonstrates that human cardiomyocytes have a lifespan of decades. However, this study cannot answer the question of whether the turnover is stem cell based or simply due to proliferation of the number of cardiomyocytes. There is some more information that is relevant in this regard. One group examined at autopsy the hearts of 8 men who had undergone heart transplant and had been transplanted with female hearts. All the cells in these hearts would have been expected to have only the female XX chromosomes. However, the investigators discovered that the cells in the heart also contain some cells with XY chromosomes, that is male chromosomes. Obviously, these cells could only have come from the recipient's body. This figure taken from the paper shows that the Y chromosomes dyed blue in the stain in the heart muscle. This proved that during life, cells from elsewhere were being recruited by the heart for regeneration. This paper, published in NEJM in 2002, created another stir and also suggested that the heart was capable of regenerating. But all this begs the question, why do we then not create new muscle to repair the heart after a heart attack? Obviously, the regenerative process which does occur does so at subclinical levels, which are unable to make any difference to the clinical outcome. In other words, the process is simply not efficient enough. The trick therefore lies in augmenting this process so that it could make a difference in the outcome after a heart attack. In the next module, we will investigate the types of cells in the heart which do regenerate and how we could try to augment the process for a good clinical outcome.